This is the last video for our genetics unit, video 11.5 on chromosome disorders. This video will cover these goals. Pause the video if you want a closer look. This slide previews the video. I'll talk about macro mutations with a focus on these four types: deletion, duplication, translocation, and inversion. Then I'll talk about chromosome imbalance, which is also called aneuploidy. Aneuploidy can be trisomy or monosomy. We'll use Down syndrome, trisomy 21, as an illustrative example of how this works. When I talk about macromutations, I'm talking about errors in big chunks of chromosomes. Macromutation errors are caused by errors in crossing over or homologous recombination in meiosis. When I talk about chromosome imbalance, I'm talking about losing or gaining whole chromosomes. Chromosome imbalances happen when there's not equal dividing of homologous chromosomes in anaphase 1, or there's not equal dividing of sister chromatids in anaphase 2. This type of error is called non-disjunction. Let's start with macromutations. Remember, crossing over is supposed to be a swap of equal chromosome pieces between homologous chromosomes. Well, sometimes that swap can go wrong. This is a huge problem when the messed up chromosome ends up in a gamete and then the gamete is fertilized to become part of a zygote. Then every cell in the zygote has this big error. The first type of macromutation is a deletion macromutation. This is when a crossing over error cuts out a big chunk of a chromosome. When crossing over, this chromosome gave up A, B, C, and D, but what it got back from its homologous partner was A, B, and C, so part D has been deleted. The next type is a duplication macromutation. This is when there are doubles of huge chromosome sections. In this case, this chromosome crossed over and gave up its A chunk, but ended up getting back an A, a B, and a C, resulting in A, B, C, B, C, D, E, and so on. Here's a diagram showing how deletion and duplication macromutations happen. The pink bands and the black bands are two different alleles at the same genetic locus. During prophase 1, the homologs pair up, but they pair up in a wrong spot. But then they still swap chromosome pieces. The chromosome on the left ends up with a deletion, having none of the locus of interest. The chromosome on the right ends up with a duplication, getting both the pink and the black allele for the same locus. Next, we have inversion and translocation macromutations. Inversion is what it sounds like. A piece of chromosome gets flipped or inverted during crossing over. So you see that A, B, C, D, E turns into A, D, C, B, E, where the B, C, D chunk got inverted or flipped. Translocation is when two non-homologous chromosomes pair up and cross over. This chromosome down here paired up with a non-homologous partner, gave up its A and B, and received M and O in return. Here's how an inversion can happen. During crossing over, one of the chromosomes got looped. The other chromosome is just fine, or is a conforming chromosome. So the crossing over points are the same, here and here, but one chromosome ends up with an inverted piece of DNA. Here, you can see that this chromosome got looped, and the crossing over breakpoints are right here and right here. This diagram shows a translocation. You can see that the red and blue chromosomes are non-homologous. They're not even close to the same size, but still they pair up and swap pieces anyway. Now, genes end up in the wrong spot on the wrong chromosome. You can see that this one ended up with a blue chromosome that was just fine. This gamete ended up with a red chromosome that was just fine, but there's translocation macromutation in those two gametes. If these two gametes end up being fertilized, then there's going to be a potential problem in the zygote, whereas these are still just fine. Besides macromutations, our other main class of chromosome disorder is chromosome imbalance. Chromosome imbalance is when a gamete has a gain or a loss of whole chromosomes. If this faulty gamete is fertilized, then all of the cells in the resulting offspring will have this problem of a chromosome imbalance. The cause of chromosome imbalance is non-disjunction. Non-disjunction is when the chromosomes don't split apart properly in anaphase of meiosis. Now, this can happen in anaphase 1 or anaphase 2. In anaphase 1, if homologous chromosomes stick together and both go to one pole of the cell and none to the other side, that's a problem. Non-disjunction can also happen because of an error in anaphase 2 if sister chromatids stick together and don't separate. Here's a diagram showing non-disjunction. On the left, you see non-disjunction in anaphase 1. These two homologs don't separate. If you follow through meiosis, you see that all of the gametes are messed up. These two have an extra chromosome, and these two are missing a chromosome. On the right, you see non-disjunction happen in anaphase 2. Anaphase 1 happens normally, but these two sister chromatids don't separate in anaphase 2. You can see that when non-disjunction happens in anaphase 2, half of the gametes end up normal. One of the 4 gametes has an extra chromosome, and one of the 4 gametes is missing one of the chromosomes. Having defective gametes isn't necessarily a problem unless the defective gamete is involved in fertilization to make a zygote. If the defective gamete makes a zygote, then all of the cells of the zygote will be affected. This diagram shows all of the non-disjunction errors that you saw in the last slide. On the top, non-disjunction happens in meiosis 1, and all of the gametes are affected. At the bottom, non-disjunction happens in anaphase 2, and only half of the gametes are affected. These gametes are normal. Then the red gametes are fertilized by a normal blue gamete, and the zygotes on the right, over here and over here, show all of the possibilities. When non-disjunction happens in meiosis 1, shown on the top, and those gametes are fertilized, you see that 2 zygotes end up with trisomy, and 2 zygotes end up with monosomy. Trisomy is when there's 3 of a certain chromosome, and monosomy is when there's only 1 of a certain chromosome. When non-disjunction happens in meiosis 2, as shown on the bottom, and those gametes are fertilized, you see 2 normal zygotes, 1 trisomy zygote, and 1 monosomy zygote. Chromosome imbalances are so devastating to a zygote that usually the zygote just dies. When the zygote dies on its own, or the fetus dies on its own, that's called a spontaneous abortion or a miscarriage. Chromosome imbalances are pretty rare in living humans. There are only a few examples, and those examples are shown here. And that's because most of the time, missing or having extra chromosomes is lethal. The most common chromosome imbalance is Down syndrome, or trisomy 21, when there's an extra chromosome 21. We can learn a lot about how chromosome imbalances work by looking more closely at Down syndrome. It's thought that Down syndrome, trisomy 21, is non-lethal because chromosome 21 is a relatively small chromosome and doesn't have as many genes on it as some of the larger chromosomes. Now, a syndrome is a medical condition with a big collection of symptoms, and among different people, the symptoms aren't consistently mild or severe. So some of the symptoms of Down syndrome are variable developmental delays, so it might be mild in some people and severe in other people. There's increased susceptibility to some other diseases, and the physical phenotypes that are shown here. You can see some phenotypes in the hands and the feet, but what a lot of people notice first about a person with Down syndrome are the facial phenotypes that you can see in this diagram and on this cute little baby over here. Down syndrome can happen because of non-disjunction errors, chromosome imbalances. You can see in this case that a non-disjunction error happened in the egg, where the egg ended up with 2 chromosome 21s. Fertilization by a normal sperm adds a 3rd chromosome 21, and now the zygote has trisomy 21. Every cell in the resulting baby will have 3 chromosome 21s. Sometimes, though, only some of the cells in a person with Down syndrome have trisomy 21. Let me explain. So you can see in this diagram that the zygote has 46 chromosomes. In other words, a normal egg and a normal sperm made this zygote, and the zygote has 2 chromosome 21s. But non-disjunction can happen in mitosis as well. So the first mitosis was fine, with each daughter cell ending up with 46 chromosomes, 2 chromosome 21s. But then after that, one daughter cell had a non-disjunction error in mitosis. The sister chromatids of chromosome 21 stuck together in anaphase. As a result, one of the daughter cells had trisomy 21. The other daughter cell has monosomy 21. That daughter cell undergoes apoptosis because monosomy 21 is lethal. But over on the right, mitosis is happening perfectly fine. What you can see at the end is that 1/3 of this person's chromosomes have aneuploidy, or trisomy 21, and the other 2/3 have a regular amount of chromosomes. This person will still have Down syndrome, but this is called mosaic Down syndrome. It's likely that this person would have a more mild phenotype compared to a person that has all of their cells with trisomy 21. Mosaicism is estimated to cause about 15% of Down syndrome cases. Then about 80% of the cases are where a person has all of their cells affected. And if you've been crafty with your math, that leaves another 5%. So what's going on with that other 5%? Well, translocation can also cause Down syndrome. If you look at chromosome 21 on this karyotype, there are just 2 copies of chromosome 21. But check out chromosome 14. Chromosome 14 had a huge translocation with a big old piece of chromosome 21 being attached. So, in one of the gametes, chromosome 14 had a mispairing and a miscrossing over with chromosome 21. So in effect, since this is such a big piece of chromosome 21, it's just like having trisomy 21. So this person would also have Down syndrome. There are a couple of other interesting things about Down syndrome that I want to share with you guys. One is that the older the mother is when she gets pregnant, the higher the chance for Down syndrome. You can see that a younger mom would have less than 1 in 1,000 chance of having a baby with Down syndrome. But a 49-year-old mom would be about 100 times more likely to have a child with Down syndrome. Now, it's not really understood why this happens, but the cool thing is, if you're interested in figuring this out and solving this problem, you could join a lab that studies this phenomenon, and you could become famous for having figured this out. Another question that pops up is to think about if 2 people with Down syndrome can have a baby without Down syndrome. Well, the answer is complicated because infertility is a big symptom of Down syndrome. Most people with Down syndrome are infertile, but let's suppose that 2 people with Down syndrome were fertile and decided to have a baby. We could do a Punnett square and see how this situation plays out. Now, because there's 47 chromosomes, meiosis would result in half of the gametes of a person with Down syndrome having 23 chromosomes, and half of the gametes having the extra chromosome, or 24 chromosomes. So, if you cross all of the 4 possibilities, normal amount of chromosomes, extra chromosome, and extra chromosome, you'd see one combination leading to a child with 2 chromosome 23s, 2 possibilities of trisomy 23, and 1 possibility of 4 chromosome 23s. Having 4 23s would be lethal, so this baby wouldn't even be born. So to answer the question, if two people with Down syndrome happen to be fertile and made a baby, there's a 2/3 chance that the baby would have Down syndrome and a 1/3 chance that the baby would not have Down syndrome. So what do we learn in this video? Well, let's recap. We learned about macromutations and chromosome imbalances. Macromutations are large chromosome errors that result from crossing over errors. The macromutations that we talked about are deletion, duplication, inversion, and translocation. Chromosome imbalances are also called aneuploidy, and that just means there's one extra whole chromosome, called trisomy, or one missing chromosome, and that's called monosomy. Aneuploidy, or chromosome imbalances, are caused by non-disjunction errors during anaphase 1 or anaphase 2 of meiosis. Anaphase 1 is more severe form of non-disjunction because all resulting gametes are affected, but non-disjunction during anaphase 2 affects only 50% of the gametes.